Our topic is nucleic acids. So, medyo magaan pa tong portion na to na nucleic acids. Okay? So, before we start, of course, lagi naman ganito siguro. Sanay naman na kayo sa akin. Before we start, let me ask you something. I launch a poll dito sa ating Zoom. So, the poll is, what is the difference between nucleic acids and nucleotide? I'll give you 2 minutes to answer this question. Again, hindi kailangan tama, no? Kung ano yung pagkakaalam ninyo. Kung ano yung thinking ninyo ng pagkakaiba between nucleic acid and nucleotide. So what is the difference, again, between nucleic acids and nucleotide? Kung hindi makita, kahit direct message na lang sa akin, no? What is the difference? Yes, pwede naman na ganoon kung nag-error sa inyo ngayon. What is the difference, again, between nucleic acids and nucleotide? Actually, yung difference na yan is na-observe na natin with other biomolecules. Yep, Maisel, pwede mo i-chat, no? Pwede mo directly message sa akin. So what is the difference between nucleic acids and nucleotide? Kapi tayo. 30 seconds to answer the question. Again, what is the difference between nucleic acids and nucleotide? Five seconds. 2 and 1. Okay. Wow, haba naman ang answer natin. So let's see kung ano mga sinagot ninyo. Okay. Nucleotide daw is the monomer of nucleic acid. Nucleotide are the components of nucleic acid. Meron pa? Ano pa bang sinagot nyo dito? Monomers, okay. Nucleotide is the building blocks of nucleic acid. Nucleic acids are polymers of nucleotides. Pa ibang sinagot dito sa chat. Tingnan natin. Nucleic acid are genetic information to new cells, while the nucleotides are repeating units. Nucleic acid is the finished product. Nucleotide is the building blocks of these nucleic acids. Nucleotide is RNA and DNA. Nucleic acid, nucleus, and presence of phosphate groups. Nucleotide is the monomer, wherein the nucleic acid is the chain of nucleotides. Nucleotides is formed because of combination of sugars. Ang haba. Okay, interesting idea. Difference po nila, may sugar. Nucleic acid is a polymer. Nucleotide is composed of pentose sugar, nitrogenous base, and phosphate group. So let's see, ano-ano ba yan? Okay, later makikita naman natin kung ano yan. Okay, so nucleic acids, no, is also a biomolecule. Ang difference ng nucleic acid with nucleotide is correct naman. Marami naman nakatama sa atin. No, baka ano na rin tayo, aware na rin, or you all have already reviewed the PowerPoint presentation. So baka naging aware na kayo, no, with your senior high school days. So nucleic acids are polymers. These are long chains of biomolecules. that has a repeating unit of nucleotides. So yung nucleotides natin are the monomers of nucleic acids. The difference lang with nucleic acids and nucleotides with other biomolecules. Mute lang natin. Okay. Okay, the difference is, for example, in carbohydrates, you have a repeating monosaccharides, right? You have a different monosaccharides combinations. For protein, you have different amino acids combinations. For nucleic acids, you have different nucleotides. But that nucleotide have different components. Okay? So yung mga nagsagot kanina na meron yung sugar, may phosphate, and merong nitrogenous bases, yun yung components actually ng ating nucleotide. Okay? Yung tatlo na yon. So hindi mo matatawag na nucleotide without the one. Without isa sa mga yon. So for this afternoon, we're going to discuss the different types of nucleic acid, its structure, and how nucleotides are formed. Diba? As I've mentioned earlier, nucleotides is the monomer of nucleic acid, and it contains different compounds, different molecules. Okay? For you to call it as a nucleotide. We're also going to discuss the primary nucleic acid structures. So, hindi lang tayo sa primary. Pag-usapan din natin yung secondary structure. Both RNA and DNA. We're also going to detailedly describe the DNA double helix, its replication, paano bang nagaganap yung translation or transcription, no, from our DNA codes? Anong portion ba ng nucleotides or ng nucleic acids yung very important in the DNA codes of our body, of our system? How our proteins are created? Diba? Bakit yung iba double eyelid? Why the others are not? Why some are brown? Okay, fair complexion, while the others have white complexion. It's all because of this, no? With these components of our nucleic acids. But again, balikan muna natin, how does nucleotide is formed? Okay, di ba? As I mentioned earlier, there are different compounds for us to create a nucleotide. So there are actually 3. We can classify those compounds into 3. So let's watch this video.
Conquer the earth in one.
So this diagram actually is a DNA. This is a DNA. Okay? And as you can see, there are different components for it to be created. So generally speaking, our nucleic acids contains these 3 main group of compounds. First is the phosphate group. Okay? This is the phosphate group. Then we have also the pentose sugar. So DNA and RNA have a different pentose sugar, right? For RNA, we have a ribose sugar. For DNA, we have a deoxyribose. Okay? Kaya DNA, kaya RNA because of that sugar difference. And we also have a part of our nucleic acid that we call a nitrogenous base or a heterocyclic base. So basically, all of nucleic acid contains these 3 compounds. The phosphate group, the pentose sugar, and the nitrogenous base or heterocyclic base. Okay? Kahit DNA pa yan, kahit RNA pa yan. Ganyan yung kanyang general structure of the nucleotides. Meron lang mga certain differences kung ano yung na kung ano yung specific type of nitrogenous base and pentose sugar yung nandoon. So let's start muna with the DNA kasi this is more common. Diba? Ito yung mas common na naririnig natin when we talk about nucleic acid. The DNA. Bakit? Kasi very special itong si DNA. Why? Because it contains our genetic information. Diba? It has our genetic information from our parents, diba? Yung chromosomes. For us to be a human, diba, kailangan syempre mag-mate. Kailangan mag-intercourse. Kailangan mag-meet yung sperm cell and the egg cell of our parents para tayo ay makreate. Okay? So in our body, kapag buo na tayo as a human, sa mga nucleus of our cells, we can find the DNA where doon sa chromosomes. Nasaan yung chromosomes? In the nucleus of our cells. Okay? So ito yung recipe kung paano tayo na-create. Diba? Yun nga, sabi ko, depende kung halimbawa bakit kulot ka, why do you have a straight hair or a curly hair? It's because of the DNA that is coded within you. So that is the general function of our DNA. It carries the genetic information. For us to say kasi na we are a living organism, we have a capacity to reproduce. And our DNA is the reason for that because we can transfer our DNA genetic information through our siblings, diba? Through the DNA. Okay? So our protein is coded based on what is inside our DNA. Okay? So aling part, no? Which parts of our nucleic acids, diba, creates the proteins or dictates what protein will be coded within us. So generally speaking, there are 3 components, di ba, ng nucleic acids. So tatlo yan. There are ito yung nitrogenous base or heterocyclic base. Parehas lang yun It's just the same. And the sugar. So it's either ribose or deoxyribose. Okay, it depends if it is a RNA or DNA. And the phosphate group. For heterocyclic bases, we can still break it down into pyrimidines and purines. So later, we will discuss about that. Okay? So RNA and DNA. So RNA are found mainly in the cytoplasm of living cells. Mainly, Hindi naman ibig sabihin, wala na yun sa ibang part. Kasi meron din yan sa mitochondria. No? Sa nucleus, actually, meron din sometimes nakikita na RNA. But it is found mainly in the cytoplasm. Meanwhile, the DNA is found mainly in our nucleus. Okay? So let's proceed muna sa general structure ng ating nucleotide. So our nucleotide have these 3 components. The image here is actually an RNA. Diba? Bakit ko sinabi na RNA ito? Kasi the pentose sugar is ribose. Ay, tatandaan nyo to Kaya deoxyribose, di ba na-discuss natin yan in carbohydrates? Because for DNA, the sugar component is missing 1 hydroxy group. Kaya nga deoxy. Di ba natanggal tong oxy na to? Pumalit ay hydrogen. Kaya naging DNA ang tawag natin doon. But basically, yun nga, all of nucleotides contains a phosphate, a sugar, and a base. Okay? These 3 components. So pag-usapan muna natin the heterocyclic bases. Heterocyclic base, I will say, is the most important part of RNA and DNA. So this is just my opinion Pero bakit? Why did I tell that this is the most important part of our DNA and RNA? Because this is the basis of how our proteins will be created. Okay? So these heterocyclic bases, as I mentioned earlier, di ba, merong pyrimidine and purine. So nitrogenous bases. Bakit siya tinatawag nitrogenous bases? Because it contains nitrogen. It is also called heterocyclic bases because it has a ring. Okay? It has a ring. This is a ring. Hindi man bilog yan, this is a ring. Kasi from end to end, connected sila. Okay? And kaya heterocyclic because it contains a nitrogen. Okay? It's a ring that contains other than the carbon atom. So normally kasi yung mga cyclic natin na molecule, kapag organic materials, walang nitrogen. So pag heterocyclic, ibig sabihin meron kang different element in your ring. So the pyrimidine contains one ring. Purine contains two rings. Okay? Again, pyrimidine contains one ring. Purine contains two rings. And these general classifications also have different na components. For pyrimidines, we have U, T, C. Okay, ano yung U, T, and C na yan? U is for uracil, T is for thymine, cytosine, letter C. The purine bases contains adenine and guanine. We have two rings for purines. So nakikita nyo, merong mga letter, di ba? Na corresponding for each of our bases. It's because the coding, the encoding of proteins in our body, no, from the DNA, from our genes, is coded through these different bases. That's why I have mentioned earlier that for me, this is the most important portion of our nucleic acid because it will dictate how our characteristics will be, no, will be coded. Okay? How proteins will be how proteins will be built. What amino acids will create us? What amino acids creates a curly hair? What amino acids create a straight hair? Ay, kung paano nakikreate yung mata natin, yung ilong, yung labi, yung tainga, yung build natin, di ba? Kung petit ka or not, it's all because of the DNA within you and the coding of these different pyrimidine and purine bases. Okay? So our DNA contains the bases A, G, T, and C. Adenine, guanine, thymine, and cytosine. Okay, this is for the DNA. For RNA, the RNA does not contain thymine. What does it contain? It contains the pyrimidine uracil. So instead of thymine, you will replace it with uracil for RNA. Okay, so here are the different heterocyclic bases found in our nucleic acids. So this is uracil. As I mentioned earlier, it is only found in RNA. Okay, then thymine and cytosine. Saan sila nagkakaiba? What are the differences? So, for example, itong uracil and thymine natin, magkaiba yung portion na to. Right? Diba? Magkaiba sila. Diyan lang sa portion naman na yan. For cytosine, iba ito. Ayan, iba yung nakakonect. Diba? Parehas na oxygen. Iba yung nakakonect dito, NH2. And iba rin dito sa portion na to. Okay, so that is their difference. For purines, magkaiba si adenine at guanine. Saan? Ayan, ito. Sa NH2, it's a different, di ba? Saka dito sa portion na to. Ayan, so those are their differences. And these heterocyclic bases, they are generally paired together. Okay, so DNA natin, di ba, double helix yung ating DNA. They are paired together. Okay? So later, papakita ko sa inyo ano ba yung pairing na sinasabi ni Sir na yan. So punta tayo on the next compositions of our nucleic acids, the sugar and the phosphate. So the sugar and the phosphate connects together. Okay? So the sugar and the phosphate creates the backbones of our nucleic acids. If we'll move back doon sa video natin kanina, asan? Medyo malayo na pala. Ito. If you look at this, di ba? This is phosphate. This is pentose sugar. They are alternately sequenced, di ba? Salitan. Because this phosphate and pentose sugar combinations create the backbone of our nucleic acids. Okay? So how are they connected? Ba? They are connected. Ayan. They are connected in the number 5 and number 3 positions of our nucleic acid. Okay? So una, if you look at this portion, ayan, number 5 carbon, nakakonect yung phosphate natin. So ba't naging number 5 yan? Dito ka mag-i-start na magbilang. So 1, 2, 3, 4, and 5. So that is why it is coined as the number 5 carbon, number 5 position. So the phosphate group is connected at this portion, at the number 5 location of your carbon. And actually, another phosphate will bind with the number 3 carbon dito in this portion. Okay? Para ma-create yung continuity of the sequence of phosphate, phosphate, sugar. Ayan. So as I mentioned earlier, it is connected in the 5 carbon and the 3 carbon of the next. So ganyan yung DNAs natin. And we call that bond between them in the number 3 and number 5 as a phosphodiester bonds. Okay? This sequence of our phosphate and sugar, for example, the phosphate sugar sequencing dictates the primary structure of our DNA because it acts like the backbone of our DNA. Okay? The alternating sequence of sugar and phosphate groups. So for this, asan yung diagram? Ayan. For example, ito. Ito yung kulay blue. This is the nucleic acid backbone which contains the alternating sequence of phosphate and our sugar. Yan. So, merong sequencing. Saan naka-sequence? Naka-connect si phosphate sa number 5 and sa number 3 carbon of your pentose sugar. So, we also call these linkages or bond as phosphodiester. 3, huwag nyo mapagawaligtad 3,5 phosphodiester linkage. Okay? Because it is connected with number 3 and number 5. So we call that as a 3,5 phosphodiester linkage. Okay? Then, for example, in our amino acids, diba kapag nakikreate tayo ng protein, yung protein backbone natin is yung portion kung nasaan yung N and C group, diba? For our amino acid. But for our DNA, yung backbone natin is yung phosphates and the sugar. Either deoxyribose or ribose sugar. Ano naman ang didictate sa kanyang secondary structure of our DNA? So the one dictating it is the bases. The hydrogen bonding between the bases. And these bases, okay, they are partners. They pair together. Yun nga lang, very selective yung kanilang pairing. For DNA, Okay? For our DNA, adenine will only connect with thymine. It will not connect with guanine. It will not connect with cytosine. For guanine, it will connect to cytosine. Again, it will not connect to thymine. It will not connect with adenine. Okay? So, ganun siya palagi. A is connected with T. Pwede ring baligtad. T is connected with A. But it will never be connected with cytosine or guanine. Likewise, for guanine, G, it will be connected to C, and cytosine will only connect with G. This is for the DNA. Okay? Bakit? Diba? Why? Why, sir, ganoon po? Because adenine creates a double hydrogen bond. No? There are two hydrogen bond between adenine and thymine. Meanwhile, in cytosine and guanine, they form three hydrogen bonding. So that is their difference. Okay? Again, for adenine and thymine, it forms two hydrogen bonds. Okay? For cytosine and guanine, it forms three hydrogen bonds. Kaya ganoon. Kaya ganoon yung pagka-partner nila. Hindi pwede na si adenine ko-connect with cytosine and si guanine hindi pwedeng ko-connect with thymine. Because of that reason. Because of that particular or specific na hydrogen bonding na nakikreate nila with one another. Okay? So the secondary structure, di ba, of our DNA is dictated by these connections, by these complementary pairs. Ayan o, pinapakita rin dito, di ba? Meron kang dalawang hydrogen bonding. Ayan, dalawa. Then for CG or GC, tatlo. And your backbone is this portion, the alternating sequence of sugar and the phosphate group. Okay? So, ganyan. Ganyan yung secondary structure of our DNA. And in turn, ito rin yung dahilan kung bakit we form a double helix. Okay? For double helix, we have two strands of nucleic acid. The blue one and the violet one. Okay? So, yung blue is 3. Yung blue is 3 to 5. Then, yung ating violet is 5 to 3. Bakit? Nakabase yan doon sa pentose sugar natin. Okay? Kung ano yung end ng ating pentose sugar. So, they are connected by a hydrogen bond existing between the base pairs natin. So, it forms the ladder-like structure or the double helix structure of our DNA. Again, remember ninyo yun, for DNA, double strand tayo. Okay, so the two, sabi dyan, is anti-parallel. Anong ibig sabihin natin dyan? So as you can see in our diagram here, the blue strand or the blue backbone is counted from 3, diba? 3 to 5. Since anti-parallel sila, yung kapartner niya na nucleic acid strand is 5 to 3. Okay? So ganun yan yung ating DNAs. And again, pinapaalala ko ulit sa inyo, G will connect with C and A will connect with T. Or C will connect with G and T will connect with A. Not the other ways around. Kaya complementary yung mga bases na yan. Kasi ganun at ganun lang sila pwede mag-interact. Dahil nga, again, because of the two hydrogen bonds and the three hydrogen bonds existing respectively with adenine and thymine, guanine, and cytosine. Okay? So, if we will draw it in this way na parang ribbon siya, ayan yung backbone. Our backbone is the alternating sequence of phosphate and sugar group. And the secondary structure is connected by these base pairs. Okay? So, nasaan? Nasaan si DNA, di ba? Nasaan yung DNA natin? Nasa nucleus. Saan sa nucleus? Nasa chromosome. So, humans, okay, contains what? How many amount of chromosomes? 46. Paano yun na-create, di ba? 23 from the sperm cell and 23 from the egg cell. Para makomplete tayo as a human. Okay? So, kapag yung chromosomes natin ay hiwalay natin, we will see our DNA. Ganun lang basta? Hindi. Because our DNA is coiled. Kumbaga, sa sinulid, no, in a thread, di ba, yung thread, it has a container. Di ba, merong container yung thread natin or yung sinulid. Nakabuhol siya doon sa container. Kung nakakita na kayo ng thread, nakabuhol yan. It is coiled to a normally white, di ba? White yun na tube. Or halimbawa na lang natin tissue, a tissue paper. No, yung tissue sa CR, for example. Di ba meron kang tube dun sa gitna? So ganyan din yung DNA natin. It has a tube inside. That the DNA is coiled into that tube. And we call that protein that the DNA is coiled in histones. Okay, we call that as histones. Ito yan. So ganyan yung DNA natin. Para organize yung DNA natin, nakapulupot yan, nakaikot doon sa histones natin. So kapag yung ating DNA ay in-stretch out natin, makikita na natin dyan yung genetic information. So the gene, each gene components, each gene characteristics. For example, the gene for example, gene for curly hairs. Nandyan yan sa loob ng DNA. So different portions of our DNA dictates different characteristics of our body. And that gene, if you will zoom in again, you will see different combinations of nucleotides in it. Okay? Kaya lagi kong sinasabi, yung 4 na yan or 5 na yan is very important kung paano tayo nakikreate. Because this is the code. Those base pairs of nitrogenous bases. Okay? So yung mga yan ang nagdidictate ng ating code. Kasi yan yung nasa inside of our genes. So chromosomes have different portions. They have 2 identical chromatids, and these portions, the short arm structure, we call that as the P arm. The long arm structure is the Q arm. The portion where they are constricted or they meet together, we call that as the centromere. So, anong difference? Minsan napagbabaliktad kasi natin yan, di ba? We use genes and DNA interchangeably. So, what is the difference between genes and DNA? So, genes actually is the individual sections. Yung DNA, yan yung buo. Diba? Yan yung pinakabuo na ating genetic information. But the genes are the individual sections. For example, diba, there are hereditary diseases. So there are gene codes for example, the diabetes. There's a gene code for Alzheimer's. There's a gene code for cancer. So there are different gene code, but it is within the DNA. So this gene code dictates the characteristics that we will have. And those characteristics is because of the different coding different sequence of proteins. And those proteins is created because of the different sequencing of different bases or nucleotides in our DNA. Okay? So ganyan kung paano na-transfer from our DNA kung paano itsura natin ngayon. Okay? Because of those coding. So kung bakit halimbawa kung ako ay ba't hindi maganda yung ilong ko, kung bakit matangos yung ilong ko, kung bakit singkit ako, kung bakit bilugan yung mata ko, is because doon sa different sequencing of the nucleotides in our genes. Okay? So gene is the fundamental units of heredity. Okay? Kaya nga, 'di ba, nanggagaling yan sa magulang. So most probably, 'di ba, meron tayong nakuha ng mga traits with our parents. So for one single organism, kaunti lang. Ba yung viruses natin, they have around hundreds of genes. For a bacteria like E. coli, it contains around 1,000 genes. For human cells, dahil tayo ay multicellular organism, we contain 25,000 gene segments. Okay? So yung bacteria, yung viruses, yung humans, magkakaiba tayo because of these genes. Di ba? Hindi naman lahat ng COVID, hindi naman lahat ng coronavirus ay kasing grabe ni COVID-19. Pero maraming coronavirus. Di ba? Kasi parang human din sila. May iba't iba silang tinatawag na strain. Di ba? Hindi naman lahat ng human mapuputi. Hindi naman lahat ng human matangkad. So iba kasi, 'di ba? Kahit pa parehas sila ng organism, parehas sila ng species, magkaiba yung gene code nila. So possibly merong coronavirus na wala namang effect masyado sa atin. Ayan, merong Escherichia coli, hindi naman siya nakaka-harm with merong mga strain yan, hindi naman nakaka-harm with a person. Pero there are also strain for E. coli na harmful for us. So the genes, okay, yung genes, yung dahilan kung bakit meron nung pagkakaiba-iba na yan. So, paano ba? Diba? Paano ba natin na-discover yung DNA na yan? Diba? Bakit ba diniscover pa yan? Nahirapan tuloy kami sa Biochem. So, DNA is discovered by Friedrich Mischer. Okay? Si Friedrich Mischer. Ano siya? Isa siyang physician. So, isa siyang doktor. Saan niya nakita? Sa pus. Pus ng surgical bandages. Saan particular? Nakita niya in the nucleus of the cell. Okay? Nakita niya dun sa nucleus. Kaya, ang initial na tinawag sa ating nucleic acids is nuclein. Okay? Kasi it is found in the nucleus. Kaya naging nucleic acid yung tawag natin. So, na-discover siya ni Friedrich Mischer. Then, si Albrecht Kossel naman, ang ginawa niya, nag-isolate siya. No, nag-isolate siya ng mga nucleic acids and tinignan niya what are the different components of this nucleic acid. So, nakapag-isolate siya ng 5 nitrogenous bases. So, pinag-aralan yung DNA. They conduct many researches and studies until such time na makonclude nila. No? Specifically, itong tatlo na to. Ni Oswald Avery, Colin McLeod, and Maclean McCarthy. Okay? Na yung DNA natin, it carries our genetic information because of the different studies na ginawa nila. Okay? So, dyan na nag-start yung mga studies. Kung paano ba natatransmit yung different characteristics, yung different diseases from one parent to its offspring. Kung paano natatransfer yung mga physical attributes or characteristics ng isang magulang sa kanyang anak. So, dyan na nagkaroon ng maraming studies after that discovery or after that conclusion of these 3 people. So, paano naman na-discover yung structure of our DNA? So, si Rosalind Franklin, nakuha niya ang isang X-ray We call that the photo 51. Okay, na-photograph niya yung DNA. Ayan, ito yan. Yung parang may pa-X na ganyan. So, nag-conduct siya ng X-ray and he found out that. Okay? Pero ang nakapagpaliwanag ng structure niyan, although na-picture ni Rosalind, hindi nga lang siya yung nagpaliwanag ng structure. Ang nagpaliwanag ng structure is si James Watson and si Francis Crick. So, sila ang nakapag-deduce, okay, that the helix structure of DNA, that the DNA has a double helix structure. And because of that discovery, na-awardan sila ng Nobel Prize in Medicine. Okay? So, kaya pa? Wala na kayong choice. So, this is replication. Ano naman yung replication? It's very important for an organism, di ba? To have its DNA. And within our cells, nire-replicate yung DNA. When we say replicate, it means it produces an exact copy of that particular DNA. By what process? So the process is replication. Replicate. So how it is done? It is done through semi-conservative replication. Sir, ano po ba ang semi-conservative replication? Halimbawa, nag-photocopy ka. Meron kang nakitang lumang examination for biochemistry. Pinotocopy mo. Di ba? Nakakuha ka ng exact copy, right? Pero hindi ganun yung semi-conservative replication. Bakit? For that case, pinotocopy mo, nagkaroon ka ng dalawang copy, na-retain pa din, di ba? Na-retain pa rin yung original copy mo. But for semi-conservative replication, hindi na siya nare-retain. Why? Because the parent DNA is being used for the another copy or the two daughter DNA. Okay? 'Di ba yung double helix natin, it has a two strands. Those two strands will be separated. Okay, will be separated. Okay? And that separated strands will be a component of the new DNA molecule. So this is our parent DNA. Yung kulay blue, mahati yan sa gitna. And it will be paired by a new strand to create a two exact copies. Okay, again, kaya semi-conservative because you use your original parent DNA to create, okay, the two daughter DNA molecules. So wala na, 'di ba? Wala na yung original mo na parent DNA kasi naging component na sila of your daughter DNAs. So there are several steps. There are 3 steps. Okay, I'm sorry, na-disconnect. Na-disconnect lang ako saglit. Andiyan pa rin kayo, okay? Na-disconnect lang ako, no? I do not know why. Okay naman yung signal ko. Unstable. Unstable ang aking connection. So, yes, noted, John Cornell. Okay na ba? Na-disconnect ako eh. Natanggal yung aking ano. Is my voice clear? Clear naman? Mukhang clear naman? Or hindi nyo, ay naka-clear naman, naka-thumbs up. It's good, okay? Ayan. So, there are 3 steps for the replication process. Okay, 3 step yan ha. So, iisa-isahin natin yung different steps of replication. Hindi yan basta tinanggal, kumabit na lang. Hindi ganoon. There are different steps for the replication of DNA. The first step is the unwinding of the double helix. Kumbaga sa lubid, no? Halimbawa yung rope, 'di ba? Yung ropes are intertwined. Pwede mong tanggalin, 'di ba, yung different components of that rope. Through what? 'Di ba? Through. Paano? By unwinding it. So ganoon din sa DNA. 'Di ba yung DNA natin, it has two strands. Kailangan muna, kasi nga semi-conservative replication tayo, we need to separate first the two strands of our DNA. And that process is the unwinding of the double helix. How? Paano? Through the use of an enzyme called helicase. Okay? Helicase. From the word helix. Diba? It removes the double helix. That's why it is called helicase. Okay? It separates. It unwinds our double helix. So that unwinding produces, starts on a point, we call that as the replication fork. So this portion is the replication fork, where the unwinding of the double helix starts. Okay? So that unwinding exposes our two strand of DNA. Okay? After that, para ma-replicate kasi yan, kailangan mo ng RNA primers. Ano yung RNA primers? Ito yan, yung kulay green saka yung kulay brown. Kasi nga, single strand na siya eh. So you need to replicate. Paano i-replicate? May co-connect sa kanya na strand. And these strands are the RNA primers. So dyan na magi-start. Okay? It will start the replication of our DNA. Anong na-observe ninyo dito? Di ba meron tayong violet and blue strand from our DNA? What can you observe doon sa primers na nakakonect sa kanila? The green one and the brown. Anong na-observe ninyo? Okay, what can you observe doon sa primers? In the primers connected in our violet and blue strands. Okay, correct, Sarah. Tama. Nishin. Ayan, tama, Nishin, no? Correct din. How about the others? Anong napansin ninyo? Ano pa ang napansin? What can you observe in the green primer and the brown primers? So meron tayong green, di ba? Green portion and the brown portion. Okay, correct, Althea. How about the direction? What is the difference in the direction? Ayun, Alexis, correct. Opposite direction. Jan Cornell, correct. Salungat po. Salungat sila, di ba? Naka other way around. Up and down, sabi ni Andrea. So tama, no? Pero ang reference point natin, tingnan natin yung replication fork, di ba? For the green one, itong nasa left side, it goes towards, di ba? It goes towards doon sa replication fork natin. Dito sa mga brown, it starts from the replication or near the replication fork away. Di ba? Pa-away naman siya. Ano pa yung na-observe ninyo? Putol-putol. Di ba? Putol-putol. Itong kulay brown, diretso yung nasa kulay green natin. So that is actually the step 2 of the synthesis of DNA segments. Okay? So mayroon na yan ay step 2 natin. After the unwinding, syempre, para matawag mo na DNA, dapat double strand siya. So our parent DNA needs to combine with an RNA primer para ma-create as a DNA siya, as an exact copy. So may dalawang portion siya, di ba? The leading strand, yung leading strand, yung may kumonek. Okay, ito yung leading strand natin, no? Yung 3 to 5. Okay, 3 to 5 natin na DNA strand. Yung kulay blue, yan yung lagging strand. For the leading strand, ang kumonek, sabi nyo nga, straight. Diba? Straight lang. Straight siya. But for the lagging strand, putol-putol. So parang yung connection ko kanina. It's like an internet connection. Lagging. Yan pa nag-lagging, choppy, robotic. So parang ganyan din with RNAs. The leading strand, the 3 to 5, is connected with the 5 to 3 RNA and straight lang siya, diretso. But for the lagging strand, ito, yung 5 to 3, okay, and 5 to 3 strand of our DNA, lagging, di ba? Putol-putol. No, putol-putol siya. It has gaps in between the RNA primers natin. Okay? So sa second step natin, kini-create kasi yung mga segments na yan. So different yung pag-create nila. Paano? Ayan. So di ba nga kanina meron kayong na-observe na doon sa laging strand is merong spaces, di ba? There are spaces. Okay, this is spaces. This is spaces, we call that as nicks. Okay, nicks. Nicks is synonymous to cut, break. Okay? So these nicks are the gaps, the breaks, butas, parang mga butas diyan sa ating nakikreate na DNA. So nakikita lang yan doon sa laging strand natin. Okay? And because meron, okay, because we have some nicks here, nicks, N-I-C-K, or breaks or gaps, we form fragments. Diba? Meron tayong mga fragments. Ito. Yung mga putol-putol na brown na yan. Yan yung mga fragments natin. Okay? And we call those fragments as Okazaki fragments. Okay? So, kapag nag-exam kayo, basahin nyo mabuti. Baka sounds like Okazaki lang. Yun ay sinagot ninyo, no? Basahin mabuti. Read carefully during exams. Okay? Okazaki fragments. From a scientist named Reiji Okazaki. Okay? So these Okazaki fragments, yan yung nasa in between the gaps. Okay? Yan yung fragments of the RNA, no, of RNA fragments na nasa pagitan ng mga gaps natin. Okay? So those are the Okazaki fragments. And of course, hindi naman po pwede that our DNA has these gaps, has these nicks. Diba? Has this incomplete fragments. So hindi pwedeng ganoon. So dyan na papasok yung third step natin, which is closing the nicks. Okay? We need to close those nicks. Paano? Paano? So, walang no, may tao sa labas. Okay, hindi sa akin. Ayan. So, we need to close those nicks. Paano? We have a primer. We call that, we have a enzyme, rather. An enzyme, ito, yung DNA ligase. Okay, the DNA ligase. So, siya yung kumo-complete. Okay, he completes these gaps. He adds nucleotides in these gaps, in these nicks, for the DNA replication or the daughter DNA to be complete. Okay? DNA ligase yung tawag natin dyan. Okay? So, ano-anong enzyme na ang na-discuss natin? Helicase. Diba? Na-discuss na natin yung helicase. What does helicase do? It unwinds. Diba? It unwinds our DNA. Then, ito hindi natin nabanggit pala kanina. A primer is added. Diba? A primer is added in our strand. Sa laging strand and sa ating leading strand, merong primers dyan na na-add through an enzyme called primase. Okay? Meron pa tayo dyan. Ito, polymerase, DNA polymerase. It adds. Okay? It connects. It connects the primer or the RNA in our DNA strand that comes from the unwinding. Okay? That is the use of DNA polymerase. And then the ligase, the DNA ligase, so don't be confused with DNA polymerase and DNA ligase. It closes the gaps. Okay? it connects the gaps with a correct nucleotides. Okay, siya yung nagko-connect ng tama na nucleotides dyan para ma-close na niya yung gaps na yan. So, yun yung use the DNA ligase. So, to sum it up, okay, the DNA, first step, tandaan nyo palagi, it is unwound by the helicase. Okay? After that, the step 2 is the synthesis, di ba? Synthesis of DNA fragments. And those DNA fragments is connected using a DNA polymerase. Okay? DNA polymerase. Then lastly, in the lagging strand, there are Okazaki fragments. There are nicks formed. So kailangan maisara yun. It needs to be closed with the correct nucleotide. So the DNA ligase joins those Okazaki fragments. Okay? So yan yung 3 general step in DNA replication. So with our technology now, DNA replication can be done actually in the laboratory through a machine or a process called polymerase chain reaction or simply PCR. Okay? Polymerase chain reaction or PCR. So what is PCR? It mimics the replication process of the DNA. Ginagaya niya. Okay? Ginagaya niya how? Chemically and mechanically. Niyang ginagawa yon. So maraming application itong polymerase chain reaction. Napakadami niya applications ngayon. So let's see some applications of this PCR. So PCR is being used in detections of mutations associated with genetic diseases. Ano pa? Detect presence of DNAs ng mga viruses, ng bacteria. Forensic, no? Meron tayo tinawag, di ba, na forensic chemistry. No, gumagamit sila nito. Halimbawa, there is a rape victim. Gusto makita kung sino ang salarin, ma-confirm na siya nga ba ang salarin. So may na-collect, for example, na sperm cells doon sa kanyang private parts. So pwedeng idaan niya with polymerase chain reaction. Bakit kailangang idaan sa polymerase chain reaction? Kasi kaunti lang yung ma-extract mo na DNA from that swab test. So ipapadaan mo yan with polymerase chain reaction, magki-create yan ng maraming copies of the DNA. Okay? So nilalagyan yan ng different na chemicals. Ayan, merong magnesium chloride cofactor, may DNA polymerase na nilalagay dyan, the different nucleotides of DNAs, okay, para makapag-create tayo ng maraming copies. So kapag nakapag-create na ng maraming copies, mas madali na kasi nating mabivisualize yung interested tayo na DNA portion of that sample. So ginagamit din yan in testing. Ako, na-disconnect na naman ako. Okay, nag-buff ako. Okay na ba? Okay lang ako. Narinig niyo na ako? Nag-buff kasi yung aking internet. Okay, dinig. Good. So, di ba there are two kind of testing pag merong COVID? Yung antigen test, saka ano yung isang tawag dun sa isang testing? Na yun yung mas pinaniniwalaan? Palagi pala dyan. Di ba dalawang test yan? Yung isa is a quick test. Parang pregnancy test lang. The other one is what it is called Correct, no? PCR, di ba? We call that the RT-PCR test. Di ba kapag you need to confirm, kasi dati yung antigen test, hindi pa siya ganun ka-accurate, di ba? Minsan nag-false positive, false negative. So you need to conduct a RT-PCR test. Di ba? So may PCR because it needs a polymerase chain reaction. Di ba? From swabbing, so swab yan, medyo painful, di ba? Pag sinuswab tayo. Konting sample lang yung makukuha dyan sa swabbing. But because of PCR, di ba? Because of PCR testing, i-replicate yung nakuha doon sa swab test natin. Then, i-confirm yan to a database of a COVID gene or a COVID DNA. Okay? I-compare silang dalawa, yung dalawa na yan, to confirm. Okay? By comparing it to confirm that you have really a COVID-19. Kasi konti lang yung sample ang makukuha. Imaginein natin, di ba? It will be really, really painful kung ilang sundot, di ba? Ilang poke, ilang swab test yung gagawin sa ilong natin para makakuha ng enough sample. Diba? It will take how many swab, how many pains. Diba? Pero kahit isang swab lang yung ginawa sa atin during RT-PCR, pwedeng ma-confirm that it is a COVID-19. So, di ba merong antigen test and the PCR test? So, mas pinaniniwalaan, di ba, yung PCR, yung RT-PCR test kaysa sa antigen test when it comes to COVID-19? Kasi nga, tinitigan na mismo dyan yung DNA nung swab sample mo to confirm if you have a COVID-19. Okay? So, kaya RT-PCR test yung tawag natin doon because it involves a polymerase chain reaction. Nagluloko ang internet. Baka dahil din sa ano, no? May disruption dito sa internet connection ko today. Baka na rin dahil walang kuryente. For a while, no, nagbabuff yung aking internet connection. Ayan, okay ulit. So, apologies, okay? Nagbabuff yung internet connection dito sa Mexico. So, punta na tayo with RNA. Di ba kanina ko pa mini-mention that DNA contains 2 strands. For RNA, 1 strand lang siya. Okay, 1 strand lang. Anong ibig mong sabihin dyan, sir? Ba't yung tertiary structure, parang may double helix din naman? Di ba? Pero actually, it's different from a DNA. Because of that reason. Because it is single. Okay? Single strand siya. Kung titingnan yung end-to-end, magka-connect pa rin yung dalawa na yan. Diba? Magka-connect pa rin siya. Single strand pa rin siya. For example, meron kang cable of your laptop, of your charger. Now, for example, you have a cable of your charger. Nagkabuhol-buhol. Diba? It gets intertwined. Pero single pa rin. Single strand pa rin siya. So, ganyan yung RNA natin. It is a single strand. Okay? So, likewise, with our DNA, our RNA also contains a secondary structure. Paano? It's also because of the connections between the pyrimidines and purines. Nagkikreate din sila ng hydrogen bonds with one another. Okay? To create the secondary structure of a particular RNA. Pero kung makikita ninyo, for this example of our RNA, wala ng letter T, di ba? Wala ng T. Kasi nga for RNAs, yung T or thymine is replaced by U or uracil. Okay? So uracil. Okay, so here are the differences, the summary of differences between DNA and RNA. Number 1, our DNA is double strand. Okay? Double-stranded. There are 2 strands. Diba? Kung makikita nyo, parang may dalawa siyang ribbon. So, this is double-stranded. For RNA, 1 strand lang yan. Okay? Ano pa ang difference nila? For DNA, you have deoxyribose. Ito, itong portion na to, iba yan. With ribose. Diba sa RNA kasi, ribose. Iba itong portion din na to. Okay? These combinations. Kaya deoxyribose and ribose. Ano pa ang difference nila? Sa DNA, meron kang cytosine, guanine, adenine, and thymine. Diba? For RNA, ganun din naman. Meron kang cytosine, guanine, adenine, wala kang thymine, you have uracil. Okay? So those are the differences of DNA and RNA. So there are 3 kinds of RNA in our body. So here are the 3 major kinds of RNA. The messenger RNA or mRNA, the transfer RNA or tRNA, and the ribosomal RNA or rRNA. So what are their differences? For messenger RNA or simply mRNA, it carries the code from the DNA. So diba, our DNA contains our genetic information. So how it is being translated into protein in our body? It needs to pass its information or its code to mRNA. Okay? This mRNA will transfer or will unstable na naman yung connection ko. And I apologize no na nagpa-fluctuate yung internet connection ko today. Okay, might be doon nga sa rotational blackouts dito sa ating ngayon sa Pampanga. But anyway, proceed tayo. For transfer RNA, again, now from the messenger RNA, okay, from the DNA, that the code is transferred to the messenger RNA or mRNA. After that, it will signal. Okay, it will signal the tRNA to transport amino acids. These amino acids will be assembled by our ribosomal RNA. Okay, again, from the DNA, okay, the codes will be transferred to the mRNA. The mRNA transfers or signals the tRNA to bring or to transport the amino acids of that particular code. Then the rRNA will assemble those different amino acids. So ganyan kung paano nakikreate tayo. Kung paano nakikreate yung protein and in turn, kung paano nabibuild yung ating physical characteristics. So first, the messenger RNA. As I've mentioned earlier, it carries the genetic information, diba, from our DNA. And mRNAs have a short lifespan. Taglit lang yan, mga hours lang, namamatay na siya. Oo, namamatay na siya. Bago pa, nire-recycle kasi siya. Kaya wala pang isang oras siya ay nako-convert na into nucleotides. Nire-synthesize kasi siya for other purposes, yung mRNA natin. So, tRNA naman, sige, rRNA naman natin, ay siya yung nag-assemble, di ba? It assembles the proteins. Okay, kung paano tayo makakreate is because of this. Because of the rRNA. So, it is found mainly, in ribosomes. Di ba our cell contains ribosomes? So, andyan yung ating rRNA. Okay, so our ribosomes contains rRNA, how many? 65. of the ribosomes are ribosomal RNA. Only 35% of its component is protein. Then lastly is the transfer RNA. Anong ginagawa niya? Nagta-transport, nagde-deliver of the different amino acids for the protein synthesis. Okay? So yun ang use. So paano ba yung process niyan? How is the protein created from the DNA? Paano ba yung coding na sinasabi ninyo, sir? It is because of the codon and anticodon. Okay? The codon, what is codon? This is the three nucleotide combinations in the mRNA. Okay, tatandaan nyo to ha. Each of the amino acids in our body is coded through 3 nucleotides. So each of the amino acid have 3 base pairs. Okay, meron siyang 3 nucleotides. Laging ganoon. So we call that the codon. Okay, the 3 nucleotides in the mRNA is the codon. Meanwhile, we also find a codon in tRNA and we call that as the anticodon. Okay? That anticodon is complementary with our codon from the mRNA. So that is how amino acids are being activated or being signaled from the DNA to mRNA to tRNA. Okay? By those 3 sequences of the codon. Okay? So again, what is the difference between codon and anticodon? The codon is found in mRNA and the anticodon is found in tRNA. So paano basically yung nangyayari in that? Ganito siya. Tatandaan nyo palagi, no? Please remember this, that each codon contains 3 nucleotides. And each codon represents to a particular amino acids. Kaya kung makikita nyo dito, if you look at these combinations, there are always 3 combinations of nucleic acids or nucleotides rather. Diba? And these 3 combinations represents a particular amino acids. For example, itong AUG. AUG means methionine. Okay? So mamaya meron tayong chart dito. I will show you a chart regarding this. So basically, ganito yung nangyayari with our DNA. So the DNA has a code in it, right? The genetic information is in the DNA. Then it is being transcribed in our mRNA, right? So yung mRNA natin, if you'll look at this, ayan, yung T, all T from the DNA is converted to U. Kasi nga yung RNA natin, it does not contain a thymine. It contains uracil. Then, paano ninyo titingnan? How will you look at the different amino acid being coded by this RNA? So, ganito lang naman siya. Bilang ka lang lagi, no? Count 3 nucleotides. So, 3 nucleotides, and so on. So, by looking at it, yung AUG kasi, it means methionine. CCG is proline. Tama ba? Proline ba yan? Then, UCU is serine. Cysteine is UGU. So, we have a chart for this. Ayan. Meron tayong chart. Okay? Paano gamitin yung chart na yan? Ganito lang naman siya, oh. O, hanapin mo yung, halimbawa, tryptophan. Tryptophan is this. So, pagtutugma-tugmain mo lang. U, G, G. Diba? U, G, G. Again, U. Ayan kasi yung U. Then G. G. Kaya tryptophan. O halimbawa naman yung methionine. Nasaan siya? Nasa A. A, then nasang block, nasa U. A, U, tapos ito may apat tayo na separates, 'di ba? A, U, G. Kaya AUG yung methionine natin. Okay? So ano pa ang tandaan ninyo? Lagi niyong tatandaan to that methionine is the starting codon. Okay, this is the starting codon. Methionine or AUG. Okay? Methionine is the starting amino acid and AUG is its starting codon. So kung papansinin din ninyo, yung ibang mga amino acid, minsan apat. Ito si glycine, apat siya, di ba? 4. It contains different 4 codons. Pero merong dalawa dyan na tigisa lang sila. The methionine and tryptophan. Di ba? Isa lang. Tigisa lang sila. Wala silang ibang codons. Tapos meron dyang codons that does not represent to a particular amino acid. Yan yung mga tinatawag natin na stop codon. So, itong mga yun. Okay? O UAA, UAG, UGA. So, these are the stop codons. Okay? So, for our amino acids, di ba? Diniscuss natin ng proteins. It can have a 3-letter code. Ayan, meron 3-letter code and 1-letter code. Okay? Ibig sabihin, pwede mong i-represent yung mga protein mo as a 1-letter code. So for example, ako, halimbawa ako ay SON. S-O-N. So S is for serine. O is for walang O. Walang O. O, wheel na lang. Walang O eh. Sige. Wheel. For example, wheel. W is for tryptophan. I for is isoleucine. Ito, I. And L. Diba? L is for leucine. So, try natin. Bigyan ng code itong WIL. So, tryptophan is UGG. Ayun, UGG. Then, isoleucine is AUU. O kahit alin dyan. So, AUU. Then, L is leucine. So, CUU. So, pwede naman pa-reverse. Puputulin mo lang lagi sa tatlo yan. So, UGG is W. Kasi nga, tryptophan. AUU is isoleucine. Kung 1-letter code is I yan. Then CUU is L. Yung leucine natin. Okay? So, ganyan mo pwedeng i-code yung proteins. Okay? How about kayo? Kaya nyo bang spell yung inyong names using these conventions? So, actually, medyo ganito yung gagawin ninyong assignment. I will give a particular combinations of codons and I have a secret code. I have a secret message for all of you to decode. Okay? Using these combinations of codons. Again merong 1-letter code for each of the amino acids. So, ganun yung magiging assignment ninyo for this week. So, any questions regarding our topic for today? I-upload ko po ba, sir? Yes, i-upload ko naman. Don't worry. I-upload ko yung ano natin. Yes, bibake ko recordings, no? Lalo na dun sa mga wala ngayon.